This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1199, recorded on March 7th, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And here in Western Mass, it is 41 Fahrenheit, 5°C, and it is blowing a gale. They're saying northwest 25, gusting 36. I think it may be even a little more than that. So if I suddenly blip out, it's because probably the wires blew down.
Here it started to warm up, and today it's one degree. So everything was frozen again this morning, and I see for the coming days, it's going to be above freezing for finally every day, which is good. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Check this out. It is 84 degrees.
Oh.
And, I would say variable cloudiness.
84 is pleasant, right?
Oh, 84 is fine. 84 is great.
What's the humidity? Is it medium? I mean, what do you feel? Do you feel a lot of humidity?
It's very comfortable. This time of year is awesome. Yeah. From March, April, and if you're lucky, most of May is awesome. April in particular, man, that's the bluebonnets. So low humidity, nice sunshine, not too hot. It's really cool. And, I've been through and cleared out all of the trash in my garden almost. I got another couple hours. Pretty soon they're going to come and mulch, and then I get to sit by and watch spring happen. It's awesome.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 39 Fahrenheit, which is 3 Celsius. And it says it's going to be snowing for the next hour. But of course, being that warm, nothing will stick. And then all next week, it's going to go up 59, 62. It's going to be really warm. And sunny for a bunch of days.
Wow.
Good.
If you like these programs, these science programs, we would love to have your support to do them. We don't do ads or anything like that. We just depend on your contributions. You can go to microbe.tv/contribute. What's up with ASV, Kathy?
Oh, a couple of little tidbits. Of course, it's going to be in July 14th to 17th in Montreal. And the website is asv.org/ASV2025. You can check out the program on the website, in particular the satellites, the plenary sessions, and the state-of-the-art lectures. A couple of deadlines I want to remind you about. The ASV Cares and Kitty Corp deadlines are April 9th. So ASV Cares is if you want to apply for a grant to help with caring for your child, either at home or grandma's or somewhere during the meeting. And Kitty Corp is on-site childcare at the meeting. That deadline is April 9th. And you can do that through the member portal because you need to be a member for that. And early bird registration ends May 12th. And in addition to registering, if you're planning to go, you might want to start looking into accommodations. Check those out.
If I came to ASV, could I just take care of the kids? or do I have to be sort of...
No, you have to be certified, and they all get security checked and vetted by their company and all that kind of stuff.
Okay,
Kitty Corp sounds like they're going to put them to work, which I'm not saying that's a bad thing, but
no, they have fun. They have fun projects.
Sure they do.
Yeah.
All right, we have a bit of news for you. Alan, did you put any of these in?
I did not put any of these in. I had a couple of things, and I came in, and there were all these news stories already in, a couple of which I had in mind, but they're already here.
First story, unfortunately, the second child has died of measles in the Texas outbreak, unvaccinated child. This is West Texas, of course. And there was death last week, first in over 10 years in the U.S. of measles. And these are in Lea County, New Mexico. I don't know if the death was there, but the main outbreak is in Gaines County. And now there are 10 cases in Lea County. Interestingly, 6 adults and 4 children, which is neighboring Gaines County.
Somebody put in this New York Times article that it says an unvaccinated New Mexico resident died.
Same thing. Yeah, that's it.
Okay. Oh, that's all the same.
Yeah. And I haven't looked, but I presume that these counties abut each other at the border.
Right. They point out that RFK has refused to come out and say get vaccinated. Instead, he says take vitamin A and cod liver oil, which is not going to stop
No.
replication. It's
And
just
too
absurd.
much vitamin A can be really bad.
Yes.
So
he says that he did say vaccines protect against measles, but the decision to vaccinate is a personal one. I don't know why he adds that on the end there. Just say vaccines protect. Why do you have to add that?
Because he thinks vaccines are bad, so he's not gonna. Yeah.
Plus, he's got to maintain his credibility with the anti-vax crowd.
Yes.
So I saw somewhere else, I don't know where, some anti-vax publication. They're all up in arms that they're shipping vaccine to Texas, to vaccinate people. And the guy said, "No, RFK no better than Tony Fauci." Unbelievable. Anyway, it's preventable, these deaths. Well,
Boy, that's a weird statement.
Yes.
That's
yes.
That's just so cockeyed in so
For
many
once,
ways.
something I agree. Yeah, RFK is not better than Tony Fauci. Damn right he's not.
Well, this guy didn't like Tony Fauci.
No,
Yeah.
right. Of course.
Like
They all
just
hated Tony
RFK
Fauci.
Jr.
but
Well,
yeah.
get him kicked out. That's fine. I have no problem with that. So the case count is over 150 now in Texas. This is from the Texas Department of State Human Health Services. So the one good thing, we're getting state updates, right? Whereas the CDC may be muted somewhat. You can go to states and see what's going on. 198 cases as of, oh my gosh, March 7th. That's a lot.
Yeah.
Most of them in Gaines, but then a bunch of other counties as well. Here's the age range is interesting. 64 children, 0 to 4 years. 89, 5 to 17 years. 34, 18 and up. 80 not vaccinated. 113 unknown vaccination status. And 5 at least 1 dose. There were 2 cases in New York City last week as well. It's not getting much press. I don't know why.
Well, it's not as big an outbreak.
It's just 2, yeah. Science.
And I'm sure we'll see these popping up around the country.
Yes, of course.
Because you've got this big outbreak going on in Texas, and people don't stay put.
Science article by Natalia Molodecki and Ron Suter, and the Switch Evaluation Sounding Board. So what a committee. The Switch. I thought Switch was a gaming console.
It's a Nintendo game console. It's a wonderful device. We've got 2 of them in the household. It's
the Switch.
Actually, no, that's right. We got rid of 1. We now have 1.
This is a very nice article which describes things happening leading up to and after the withdrawal of OPV type 2 from the trivalent poliovirus vaccine back in 2016. The type 2 component was causing a lot of polio, vaccine-associated polio, and there were no more wild-type cases. So they said, we should just withdraw it. And they did, but the problem was vaccine-derived polioviruses were still circulating. And then as vaccination dropped, right, because there's no more type 2, you got big outbreaks. And they give a number here, which is over nearly 4,000 cases in 3,300 children in 53 countries were paralyzed by circulating vaccine-derived poliovirus type 2, which they say is all completely preventable.
So those 4,000 cases, we're talking all paralytic polio.
Yeah, vaccine-derived, but just paralytic, it's polio. You don't even have to say vaccine-derived, but you make the distinction because it could have been avoided, right?
This, I agree with you. This article is sort of a nice, succinct, and very clear description of the whole problem. It's really very well done.
Yes, it is.
The whole goal of the switch was to stop the use of Sabin 2 to prevent VAP. However, since the switch, there's been a tenfold increase in VAP caused by type 2. And they go on to say that IPV does not cause polio, and we should be moving towards that, but it's not such a simple situation. But they're also planning to withdraw bivalent OPV. So in many countries, types 1 and 3, which are combined in a bivalent, are being used, and they want to withdraw that. But it's the same issue. There's circulating vaccine-derived viruses. What's going to happen? If you're ready
I think
with
we know
IP
exactly what's going to happen based on the type 2 experience.
Exactly right.
So the problem. So just to review,
Yeah.
these are oral vaccines, live attenuated, and the inactivated guy is an inactivated virus. That's not oral; it's injected. So
Right.
the problem with using that is mostly that it's injected and it costs more. Is that right?
Yeah.
Yeah.
The injected issue is the big issue because you need all of the stuff that's required to inject a vaccine, which includes somebody who's got at least enough training to use the needle and put it in the right spot. And you need to be able to, supply that and have the trained staff.
So it's not just the cost of the material itself, but
Right.
all of the peripheral stuff.
The whole, yes, it's an infrastructure.
That
issue.
makes sense. Yeah.
And both of them have a cold chain issue. Is that right?
Yeah.
I would
Yes.
assume
believe
so.
so. Yeah.
Yeah, so that actually has been solved rather, amazingly for the oral vaccine. But delivering the oral vaccine is you need to get a kid to open their mouth and put a few drops on their tongue, which you can train anybody to do in 15 minutes.
They point out that the expanded use of IPV, especially with a hexavalent vaccine that includes other immunogens, could be a game changer. But as with every vaccine, to harvest the full potential, high vaccination coverage will be critical, including in the most difficult parts of the world.
And most of the vaccines that we have, the whole standard childhood series is injected. So this notion of eradicating poliovirus because it's an oral vaccine it's kind of an, one-off. Like, okay, you can control poliovirus with this oral vaccine, and that's great, but that doesn't get you anything. The systems you build to do that don't get you to a point of being able to vaccinate the world against measles, mumps, rubella, diphtheria, pertussis, tetanus, yada yada yada. And so if we could bring ourselves politically to make the investment in the infrastructure to deliver injected vaccines worldwide, I don't like the cliche game changer, but it would make a huge difference. We could do all sorts of good.
So one more thing, they make the statement, polio eradication, so poliomyelitis eradication, requires the removal of all polioviruses from populations, which we've said many times here, can't happen because many countries use IPV and polioviruses are circulating in those countries. So you cannot eradicate poliovirus, but you could, if you immunize enough people, eradicate or eliminate poliomyelitis. But you can't use OPV because it causes the disease. And that's a little bit of a conundrum there. Anyway, it's a good article if you can get to it.
Yeah, it's not paywalled. Science, you can read. I think you can read something like 5 articles before they pop up a paywall. And by the way, pro tip, you can clear the cookies and history in your browser, and then you can, go to sites that blocked you out because your cookies are now gone.
My goodness. That's a good pro tip. I like
Cookies, huh? Cookies.
Yeah, I have my browser, my Firefox browser is set to purge all cookies in history as soon as I quit. Every time I fire it up. The one downside of that is it logs you out of everything, but whatever,
Article at polioeradication.org. Humanitarian access improves quality of polio vaccination campaign in Gaza. It's reached 600,000 children with better access, conflict avoidance or whatever. which is good.
Yeah.
But of course, they're using OPV in this, and so they're just seeding new outbreaks.
I'm sitting here channeling Rodney King. Can't we all just get along?
Get along.
Apparently not, Rich.
Yeah.
So.
Yeah, and, you know, in defense of the use of OPV in a context like this, or in poor countries where we don't currently have the infrastructure, we are preventing lots of kids from getting paralyzed, and that is a good thing.
Yes.
But my feeling is we just shouldn't make that the end game. You know, okay, OPV, emergency situations like this, fine, use it because that's all you can do. But the goal ultimately should be get injected vaccines into these kids everywhere.
Article in IAVI, a nonprofit science research organization. What does it stand for?
Isn't that
Rich should know.
International AIDS Vaccine Initiative? It used
There you
to be,
go.
but they've broadened their focus now.
Yeah. IAVI is the way they pronounce it.
Yeah, IAVI.
Right. First participants vaccinated with IAVI's Ebola Sudan vaccine candidate in Uganda amid Ebola outbreak. So they're doing a ring vaccination trial because we don't have an approved Sudan vaccine. So this is part of the approval process here.
Yeah, so I don't know if this is I had a TWiV pick last week that was related, if not this. But the cool thing about this is that they were ready.
Yes.
You know, this is what pandemic preparedness ought to look like.
And IAVI, they made the vaccine, right? Or they funded them.
They funded it.
They funded it, and WHO is contributing because, you know, the U.S. is dropping the ball. But normally we would help a lot with this. This is highly unfortunate. And here a story, I guess, close to me.
Yeah, so Laura just came in and showed me this on her phone, so I looked it up, and here's one of the many news stories that just came out minutes ago, I think. Trump administration has canceled $400 million in grants for Columbia University. They're claiming that due to the school's continued inaction in the face of persistent harassment of Jewish students. And this is just total BS. It's an excuse to to jerk people around.
Well,
more shock programming.
The Department of Education is investigating Columbia for anti-Semitism, right? So I walked in to teach class last week, and I saw the school newspaper, big headline on the front, and so the university is trying to address what's going on, and also Columbia gets a lot of grant money.
Yeah.
From NIH too. So they could lose a lot. Right. The news is just really not good these days.
I try and keep my distance.
Yeah.
Well,
a respectful distance. You got to know what's going on.
Yeah, you have to know what's going on.
But I can't sort of stew in it.
No.
Well, I read Helen Cox Richardson every day in the morning while I'm drinking coffee, and that sets my tone for the day. Right?
Yeah, unfortunately.
Gets me stewing.
Lots of times it's not real happy reading.
However,
I don't read I have stopped reading news in the morning with my coffee. I don't read news until after I've gotten just about up to lunchtime usually. Because I realized there's not... if there's a fire, I'll know. If there's some other immediate thing, I'll know about it. There's no urgency in my day to know exactly what's going on or what new horrors have been visited upon the world by the current administration. And I'll get to it. And then I read my news feeds, and I pared my news feeds down, and any headline that mentions Trump administration something, I just scroll past it. Unless it's about a science agency, and I need to keep up with that. So for professionally, I need to follow those particular items. But other than that focus, my feeling is, I'm upset about all this, but there's nothing I can do about most of it right now. So I'm not going to read stories about things that are horrible that I can't fix. That's my coping mechanism.
Look, the good news is last week the Supreme Court said to Trump, you can't freeze these USAID funds, which include many funds for infectious diseases,
public health,
public health. So I'm just amazed that happened. 5-4.
5-4.
5-4.
Amy Coney Barrett and John Roberts.
John Roberts
went over to the dark side.
I mean, it's congressional approval. Congress, the president can't touch it. Okay, but so the Supreme Court said you got to pay it, and now I'm just waiting for them to say no because that's the way they are.
Yeah.
Hopefully, some money will start going out again. Okay, we have some papers today for you. The first one, Kathy, can you summarize that for us?
Sure. The title of the paper is Bat-Infecting merbecovirus HKU5-CoV lineage 2 can use human ACE2 as a cell entry receptor. So they shorten it to HKU5-CoV2, which I think right off the bat might be a little bit confusing. But I'll go into that in a minute. The paper has 4 first authors, Jing Chen, Wei Zhang, Yang Li, and Chen Liu. And the senior author, there are 6 corresponding authors. Is that what we decided? And the most senior one is Zhengli Shi. And so she used to be at the Wuhan Institute for Virology, and so a lot of those co-authors are from there. And she's now at this other institute, which is the Guangzhou, right?
Yeah, so the
Guangzhou National Laboratory in Guangzhou.
And the other institutions involved: Wuhan Institute, Guangzhou National Laboratory, Guangzhou Medical University, Wuhan University, University of the Chinese Academy of Sciences in Beijing, Guangdong Academy of Sciences in Guangzhou, and University of Science and Technology of China in Hefei.
Okay, so to summarize basically from the title again, they went in a survey of bats, so they got anal swabs from bats and looked at sequences, particularly by using PCR primers for the very conserved RNA-dependent RNA polymerase, the molecule that replicates the RNA. And they identified 5 sequences from bats that were similar to a 6th sequence that was already existing. And basically, from this paper, they're going to start dividing that lineage of the bat viruses. Okay, so they call it HKU5-2. And the previous existing one lineage with other viruses in it is HKU5-1. So just to set the stage a little bit, we know of 7 human-infecting coronaviruses. And this paper isn't really going to be about any of those, but the original SARS coronavirus, SARS-CoV-2, and then the MERS. And this is somewhat related to the MERS human-infecting virus. And then the 4 so-called common cold coronaviruses, one of which has HKU in its name, but that's just because that's the, I think, Institute, Hong Kong University, where they were probably isolated. Okay, so HKU5-2 is the subject of this paper. They isolate 5 more sequences from it, and most of the work they're going to do was with the one that had been previously known. And basically, they show that these viruses use the ACE2 receptor, which is different from all of the other merbecoviruses. So MER comes from the MERS virus, and so that's how these are related to that human MERS virus. And so the fact that this lineage 2 can use this human ACE2 receptor to get into cells, and then it can grow in cell lines and organoids, and they even isolate. So first of all, they get this all just from sequence. And then they actually isolate the virus, and they do experiments with that. And it's really important work, and it shows that this is something we need to be concerned about with respect to pandemic preparedness. And the fact that it uses this ACE2 receptor is interesting because it binds to the ACE2 receptor in a different way. So they do some nice structural biology to look at that. And then in the end, they unfortunately, but evidently, they were somewhat lucky because they did this at biosafety level 2, and at least to our knowledge so far, there haven't been any bad infections of humans as outcomes from this. But we'll talk about an op-ed that or an essay in the New York Times that suggests that this work should probably have been done at a higher biosafety level.
Yeah, that's why I picked this paper, is because it's been in the news and thought people might want to know what the science is behind it. So
Yep.
we'll explain it to you. So MERS is a member of this merbecovirus, for which there are also bat merbecovirus. There's lots of them out there, right? And so this paper is addressing the question: are there some bat merbecoviruses that pose some pandemic potential? And they focus on receptor binding and fusion. Remember, the spike of these coronaviruses binds the cell receptor. There's a receptor binding domain. And then that binding actually, and then the fusion, which happens next, requires cleavage, proteolytic cleavage, cutting, like with scissors, of the spike as well by proteases in the cell. And so they look at those two issues here. So they start by getting bat anal swab samples, and then they do PCR targeting the polymerase, look for some hits, and they end up getting 5 full-length genome sequences that look like a previously identified bat merbecovirus. All right, and so phylogenetic analysis indicates that these, as Kathy said, they called them HKU 5-CoV. They're a bit different from all the other known bat merbecoviruses that seem to have a distinct lineage, and that the RBD is less similar. So they think there's a distinct binding mode here, and that's what they're going to study in this paper. So the first thing they do is they take the spike protein of BT-HKU5-CoV-2. What do you want to call it?
Can we come up with a shorter...
5-2. Just call it 5-2.
Yes, thank you.
So 5-2, which is one of the sequences they identified. They take the spike and they put it into VSV, so that's called a VSV pseudotype. So you can do this at BSL2 containment. And you see what kind of cells they infect, which means the spike is allowing entry into those cells. Of course, the VSV is doing the rest, but at least you know. So 5-2, and then they have a bunch of controls like MERS and SARS-CoV and 5-1, which is the earlier sequence, that if they overproduce ACE2 in 293 cells, human 293 cells, it effectively enters the cells with these pseudotypes. They have to do trypsin treatment, which cleaves the spike, which means there's no cleavage happening in those cells, but at least it can bind and enter. And if you knock out ACE2 in another cell line called HUH7, it abolishes entry, and then if you reintroduce ACE2 into those cells, it restores it. So it looks like 5-2 can you bind human ACE2, and it's a functional receptor for cell entry.
And seems to prefer ACE2 over the other receptor offerings in this experiment.
Yeah. Okay, so what about, so that's human ACE2. What about other species? So they got 54 ACE2 orthologs from non-human primates, other mammals, and birds. And they say most mammalian and avian ACE2 orthologs support efficient entry of 5-2 pseudotypes. So they could have a broad host range. You don't know because
this is still the pseudotype experiment. We're not using the real virus.
Yeah, it was only the binding and entry. So the subsequent steps, we have no idea.
But
we'll
this
get
is
to that.
exactly where you start with something like
You
this.
have to start with this, yeah, because it's safe to do.
Well, not only that, but it's the first step. If you're not
yeah.
going to if you're not going to attach the receptor,
No, it's fine.
game over.
Yeah.
All
Yeah,
right.
go to the next virus.
You don't want to waste time. And also, they find binding to various bat ACE2 orthologs. Okay, but it's interestingly, it's resistant to, it doesn't bind ACE2 from the bat where 5-1 came from. So 5-1 again was isolated a number of years ago, and so obviously it's different from 5-2. So maybe a broad tropism, maybe it can infect a lot of different species out there, but we don't know. And they look at the affinity, the binding affinity between the receptor binding domain of 5-2 and human ACE2, and they find that it binds very well. The affinity is 6.55 times 10 to the minus 6 molar, which is pretty good affinity. But it doesn't tell you anything about infection again, because maybe 10-fold less or 20-fold or 100-fold, maybe it would still work. So I find these affinity measurements not really informative. But that's just my 2 cents.
But again, it's a starting point. These are the things you
okay.
do first to see if this is worth drilling deeper into.
They solve the cryo-EM structure of the 5-2 RBD, receptor binding domain, bound to human ACE2. Okay, very nice. And they see that the interaction between the RBD and the ACE2 occurs in 2 patches. And these seem to overlap with the way other viruses bind to ACE2, but it has a distinct binding mode to human ACE2 compared to other ACE2 binding coronaviruses.
I just have to say this sentence. The cryo-EM single particle structure of the complex were resolved at 2.96 angstroms, period. This used to take years.
Yes.
If you were lucky at all. And now it's just, oh, yeah, sure, we solved the
I have
structure.
still not gotten over how this is just something, something we did on the way.
Oh, yeah.
Just like we're driving past this and,
We'll do that
so
after lunch.
Structure.
Cryo-EM is amazing.
Hold the elevator. I'm going to
Well, you remember SARS-CoV-2 spike, Jason McClellan did it in 6 weeks, right?
Yeah.
Beginning of 2020. Amazing. Okay, so what are the amino acids in the RBD that bind human ACE2? They change the amino acids, all right, which is called changing amino acids, not mutagenesis of the amino acids. It's changing the amino acids. It's okay. They call them mutations. I've given up. I've given up all kinds of things in the virology literature that I give up on, but you will still hear me complain. Interestingly, so they know the amino acids that contact ACE2 in the spike, right, from the cryo-EM, so they can change them. They have 1, 2, 3, 4. If they change any one of those, it only reduces binding a little bit, and it reduces a little bit of pseudotype virus entry. But if they change 2, then you have an effect. All right, so then they substitute the residues on ACE2 that from 3 bat species that don't work to bind this virus. And they put human ACE2 into it. And they find that one substitution, which I'm not going to tell you because you're not going to remember, enhances binding and pseudotyped entry. And some of them don't. In the end, they find that one amino acid, E37, is a critical residue. And they say, this bothers me a little bit, okay? They say it highlighted the well adaptation of 5-2 to human ACE2. Okay, so first of all, well adaptation is not right, but we won't dwell on that. It's not adaptation because this is not passed through humans to adapt it. It's in some other animals randomly mutating, and it just happens to have reached a sequence that does well with human ACE2. Because as far as I know, there's no evidence that this virus has ever infected humans, okay? Do you guys understand what I'm saying?
I see what you're saying,
sure.
Yes.
It's not adaptation. It's adapted. It's adapted already because of other things going on, but it's not adaptation. Okay.
To me, all of this stuff, I presume, goes into, I don't know that it's organized, but it will be, a giant database that in the long run is going to make it so that you can look at a coronavirus spike protein sequence and know what it's going to do.
Maybe, yeah.
Which is, the kind of thing you need to do surveillance.
All right, so next they isolate an infectious virus of 5-2 from a bat anal swab sample. This doesn't happen many times. I remember maybe one other paper where those banal viruses from Laos, they got an infectious virus.
And it's important to point out that everything that we've talked about so far did not have infectious virus involved.
Right.
Okay, there's a tremendous amount of the stuff that has been done with the coronaviruses that doesn't involve infectious viruses at all, and I think there's a lot of people who don't understand that.
Yeah, doesn't involve infectious intact coronaviruses. The pseudotyping that's done here, it's a virus. It can get into cells, but it's a hothouse flower. It's a lab construct of a harmless virus that's got the receptor for the virus you're studying. It's just a model system. It's not going to get out. There are a couple of reasons why it's done this way. There's the biosafety issue, but also there's the just practical issue that we know how to grow VSV. Those protocols are all really well worked out. We know what cells it grows in. And you can just take the sequence from your computer database and pop it in there and do those experiments. We don't know how we're necessarily going to culture some virus we've never cultured before, so that's a lot of work that you would have to do. You don't want to do that unless you know it's going to be worth the trip. So now we're at the point in the paper where they actually say, okay, it's worth growing up this virus.
This is interesting enough, so let's grow up the virus.
Yeah.
So they take some of... Go ahead, Kath.
I was just going to say, so you remember that there was already one virus that was in this group, and that's what they did. Used the sequence and did all this VSV work and so forth with. And then the other 5 sequences they found, one of those was the same as the one that they made, or that they were able to isolate the infectious virus from. Right. That's all I wanted to say, is that so it's still within that same group of original bat anal swab samples, and presumably they knew where they had come from, and they went back to the bat tissue and got the virus.
Yeah. And the virus that comes out of cells, they put them in CACO2 cells, which are human intestinal cells, and they have ACE2 in those. It causes cytopathic effects. It kills the cells. You could stain it for viral proteins. You get an increase in viral load. And they sequence that virus, and it's the same as the original sequence that they had pulled out of the anal swabs. We'll say now they have infectious virus. And you might say that before doing this, they may have increased the containment. That's one of the contentions that people have been making. But this is all done under the same containment. We'll come back to that later. If you knock out ACE2, the virus doesn't infect cells. If you put human ACE2, if you put human ACE2 back, it restores infection. You can block infection with an antibody to ACE2. You can block infection with SARS-CoV-2 and SARS-CoV receptor binding domain proteins that you've made, but not MERS-CoV RBD. Now, MERS-CoV RBD binds to NPP4, a different receptor. DPP4, dipeptidyl peptidase number 4. So this is what's unusual about this. It doesn't bind DPP4. And so basically, this virus that they got out is binding to human ACE2. Now, the other step besides binding is the spike has to be cleaved in order for fusion of the viral and cell membrane to occur. And there are 2 ways that this can happen for many coronas, SARS-CoV-2 for sure, but also MERS-CoV. There's a cell surface protease called TMPRSS2, which can cleave the spike at the plasma membrane, and then it will fuse. The virus will fuse with the plasma membrane and put the RNA into the cytosol. Or in cells that don't have cell surface TMPRSS2, the virus is taken up into endosomes where there's a protease cathepsin L that cleaves the spike, and then the virus can fuse in the endosome and get out into the cytoplasm. So they look at a whole bunch of protease inhibitors to look at what's going on here. They find that 5-2 can be activated for fusion by both temperas and cathepsin L. Similar to MERS and SARS-CoV-2, the 5-2S has a furin cleavage site. A furin cleavage site. It's been out there in nature circulating. Nobody ever touched it or put it there. It's so infuriating, people who say the furin cleavage site of SARS-CoV-2 is a smoking gun for lab manipulation.
Nope.
Oops, I know who said that. But it's not, because you just didn't have enough data. Anyway, so it looks like the cleavage is done by similar proteases, which is very interesting. You know, we do have inhibitors of these two proteases, so at least therapeutically they could be used. And then they say, okay, what about human respiratory and enteric organoids, right? Little cultures that, well, they're not little, cultures that resemble the respiratory tract and the enteric tract. So this virus was able to reproduce in both the respiratory and the enteric organoids. So who knows, maybe it could infect both the lung and the gut. And then they say, well, what could we do as a therapeutic countermeasure? They make a bunch of monoclonals and peptides, and they find some peptides that will suppress infection. And then the antivirals nirmatrelvir, remdesivir, and GC376, which work against many coronas, that will inhibit this one as well. So there you go. That's the story. And those last few experiments, growing up virus, that's what people are worried about. And we can talk about that.
So they, if you scroll all the way down through this, this one was paywalled, right? Mm-hmm. This was the one I had to get a copy of. So their biosafety statement, all of this was performed in a BSL-2 plus negative pressure environment, which is not there aren't specific standards that I know of that define a BSL-2 plus negative pressure environment. This is something that may be in Chinese regulations, maybe in their own institutional regulations. Negative pressure is where you have the lab at a slightly lower atmospheric pressure than the outside rooms. So if somebody opens the door of the lab, the air rushes into the lab instead of out of the lab. So this is essentially a BSL-2 with a slight change in the air conditioning system, and they keep the door closed.
So it does not prevent people working with the virus from getting infected.
Right.
It may be a little impediment to it getting out, but those people working on it could get infected and then leave and bring the virus with them. Right?
And this is in contrast to a BSL-3, where people would have, there's a kind of an airlock entry. It's also under negative pressure, but you're also in a bunny suit with mask and gown and hood. It looks a little like a clean room type of environment where people build electronics. And then, of course, there's the BSL-4, which is the Hollywood bubble suit lab, or, like when we toured the Needle. And that's the whole, containment of everything type of environment.
BSL-3, are you also wearing a mask that's connected to a filter, or is that?
Yes.
Yeah.
Yes, right.
Papper.
Yeah. So there's a New York Times, what did you call it, Kathy?
It's called the guest essay.
Guest essay.
Said it's by Ralph Barrick and Ian Lipkin, or Lipkin and Barrick in that order. And it says, recent virus research should raise the alarm.
I presume, as usual, that the headline was not written by them. It was written by the Times, okay, which is
although
reproducibly
from my experience, Amy and I wrote an op-ed a couple of years ago Yeah, they go back and forth because you make a title and then they fix it, they change it, and they say, "Is this okay?" And you can make little tweaks. You can go
Sure.
back and forth. So it's not like they just throw something out there. Although
It's
I
not
don't know
like
if
what they
it's
do to their regular staff reporters.
yeah, that's right. That's right. So they summarize the experiments we just talked about, and they say the Wuhan Institute of Virology, where many of the researchers work or have worked, is at the center of the controversy regarding the origin of the COVID pandemic. We do not imply that the institute is responsible, nor do we have any certainty that this virus can be the next pandemic virus. What worries us is the insufficient safety precautions they took. And they talk about BSL 1, 2, 3, and 4. Although, let's see. I don't know. Did they recommend something at the end? I don't think so.
Well, interestingly, if you look at the paper that we just discussed, they refer to 2 preprints that came out while they were doing a study
Yes.
that report similar findings.
Hmm.
One of those 2 preprints has as a co-author Ralph Barrick. And if you look in the methods from that paper, it is apparent to me that the live virus work that was done in that paper was done at North Carolina, where Barrick is, and they have a biosafety statement there that says it was done at BSL 3.
Okay.
So I take away from that Ralph would recommend BSL 3 for this work.
I don't think it's unreasonable, right?
No, not at all. It's not,
Because even at the beginning, before you start researching the virus, before you even recover it from these anal swabs, you don't know what the properties are going to be. So I think it's reasonable to default to BSL 3.
Absolutely.
Yeah.
I got no problem with
I
that.
agree completely. And when I saw in the show notes the editorial, and I read the paper in the editorial, I was like, yeah, I'm sure some people would call for BSL 4 here. I do not think that's appropriate. BSL 4 is a huge impediment to research. There are only a few facilities in the world. It's a whole big thing to get in and out. It really limits what you can and can't do and slows everything down and drastically increases costs. So the step up in complexity from 3 to 4 is enormous. I think most Research 1 universities have BSL 3, probably. It's pretty common. It's not cheap. It does cost more than a BSL 2. You do have to be trained for it. There are protocols. It's annoying to get in and out and all that. Got to be a special room. But it's totally doable. It's something I'm sure WIV has. It's something I'm sure these other institutions probably have. And I think that would have been a very appropriate containment level for this amount of work.
I mean, the work that was being done at Wuhan Institute of Virology pre-2020 on SARS-1-like viruses was also being done at BSL-2. You can read the papers, and they say it's BSL-2, and Ralph Barrick has been critical of that, which does not mean that led to the COVID pandemic.
No.
Because it's a totally different virus, okay? Yeah. But just in principle...
it's one of the... I'm sorry.
I think that 3 would be reasonable. I don't understand why they would not do that.
I wouldn't work on SARS-1 into BSL-2.
No.
That'll kill you. No. I mean, I'm going to want the respirator and the...
Yeah, of course.
So, these practices that apparently are SOP at Wuhan is one of the things that gets cited by those who favor the lab leak thing as an issue. I would say, it's an issue, but not necessarily an issue that speaks to the lab leak thing. Okay, those are separate deals. But there are the virus doesn't recognize international boundaries, and so we ought to have international guidelines for safety.
I'm trying to think if there ever have been other viruses isolated that were novel and that could reproduce in human cells, right? In the years before BSL 3 and 4.
Well,
but we just isolate human viruses, right?
Actually, now that you speak of it, in cases where there's been a novel virus that infects humans from some animal reservoir, we've isolated it because it infected the humans.
Yeah.
Right?
Yeah.
I mean, the first thing that jumps to mind is Lhasa. Okay. There are plenty of other examples. Okay. So this is a new level of thinking, and an appropriate level of thinking, right? The idea is to try and anticipate where the next pandemic is going to come from. So these are appropriate experiments, and contribute to that whole thing. But we need appropriate guidelines.
So the problem is that there's no international standard.
Exactly.
Every country does their own thing.
Exactly.
There are international standards for what constitutes BSL 2, 3, 4, and 1, but there aren't standards for what you do in those labs. I think there are kind of guidelines, but they're not they don't really cover a case like this because you don't know that this can infect humans. But you're looking for something that can infect humans, and the whole reason you're growing up the virus is because you have preliminary evidence that it can infect humans. If you didn't have that, you'd have gone on to do some other experiments. The other thing I wanted to mention here is I would say no matter where you are, these experiments should have been done in a BSL-3. But in particular, if you or your collaborators are at the Wuhan Institute of Virology, which has gotten so much heat and scrutiny over biosafety, not saying that's fair, not saying that it's based in reality or rationality, but given that your institute has gotten that kind of flack, you need to kind of set the bar higher. You need to say, we've got to set the example and show the world that we're careful and do these at BSL-3, please.
So go ahead,
Kathy.
I was just going to say, so what Barrick and Lipkin say in this guest essay is that, first of all, as the leading international public health agency, the WHO should take the lead in rigorously characterizing these standards. So what you just said, Alan, about, there are some standards, I think that, for instance, different agents are classified at different levels, but then the facility in which you work on those takes into account the classification of the agent and the work that's going to be done and so forth. I mean, Vincent can speak to this because he was on the biosafety committee at Columbia. So you could designate, if something is recommended to be done at it's a BSL-2 agent, but you think that it's your committee, your local committee says, yeah, we want you to do this at BSL-3. They have the ability to do that. But even if there are these international standards, there's no stick. If somebody wants to go out and do this at whatever level they want to do it at, that's one of the problems. So, but as they say, the WHO should take the lead and rigorously clarify these standards. And maybe they need to add something specific, like if you're going to be doing something from an animal that has shown crossover in the past to a zoonotic virus that has caused an epidemic or a pandemic, then maybe you ought to consider at least doing it at BSL-3.
I want to make the additional point for a lay listener that might be freaking out about all of this, that because this virus can use a human receptor and infect human cells in culture, and including respiratory cells and gut organoids, that does not mean that it's going to infect humans. There are dozens, at least, more barriers to a virus that can infect humans and be transmissible in humans, okay? So, in fact, in a way, it's not completely insane that you would do this at BSL-2, okay? Because the likelihood that this is going to be a problem it seems low, but I'd do it at BSL-3.
So, given now that we know it infects human cells, should they be doing work at BSL-3 with this virus?
Yes.
Yeah,
Yeah.
I think they should at least switch, right? Yeah.
It came from bats.
Yeah. Yeah. So, but FYI, WHO does recommend containment for different viruses. So last year they reduced SARS-CoV-2 from BSL-3 to 2. And then each country has to approve that.
Because you can vaccinate everybody.
Right.
Yeah,
Right.
you can vaccinate everybody. And the CDC approved that. So now in the U.S., you can theoretically work
Right.
with SARS-CoV-2. At the beginning of the pandemic, at my institution, which shall not be named, there were people who did not want anybody working on the virus at all, even in a BSL-3, because they thought this was before there was an outbreak in New York, because they said if there's an outbreak in New York, they'll blame us. Right? They'll say it came from you.
And again, the perception is part of the issue here. It's not just that the science points toward containment. It's that for your institution's sake, for the sake of virologists everywhere, you need to show the public that we're trying to do the right thing.
Yeah. All right. One last point about this paper. Okay, now we know this virus can infect human cells. What do you do? What do you do with it?
Yeah, exactly. I mean,
What do you do with that knowledge, which is the whole point of it?
well, you need, I don't know, maybe you just, what's the next step in trying to figure out whether it really has potential to wreak havoc in humans?
I think you don't know,
I don't know.
aside from infecting humans. But I think you need to be ready. The antivirals work, okay, so that's done. But do we make monoclonals? Do we make vaccines?
Do we test in various animal models?
That's
Put
my
it
thought.
in ferrets?
My thought.
You could.
You
If
could
it
test in animal
transmits,
models.
Yeah.
But yeah, so what if it transmits among
You still-
ferrets?
yeah,
I don't
you
know what
still-
that means
yeah.
for people. I just don't know what it means. But you have to do it. I agree,
Yeah.
you have to do it. But that's the whole point of this pandemic searching, that you have to do something with the viruses you find. And
Well, in the case of, it's relevant that they tested a couple of coronavirus drugs and they work.
Yep.
Okay, so that's
Yes.
important.
yes, that's good.
It brings to mind the news article that we had that described that IAVI had a vaccine ready for the Sudan virus. Okay,
Right.
so depending on how concerned you are about the risk, and maybe that involves some animal models or something like that. Maybe you prepare a vaccine. They did show that one of the things they showed was that a lot of the existing anti-coronavirus monoclonal antibodies don't work. Okay,
Mhm.
so that you would have to take some sort of different vaccine approach, or at least have a different antigen. The path is not clear.
Yeah.
But you need to do these experiments and get it to at least this point, where you have that list of, okay, these are viruses we found in bats. We're out doing the surveys. These are viruses we found in bats that have the potential to replicate in relevant cell types, human cell types. Here's what we know about them. Getting this amount of information about SARS-CoV-2 at the start of the pandemic took us, a month and a half. We're already a month and a half ahead if this thing crosses over into humans, so that's a nice start. And just, keep an eye on it. Maybe set up a quick test where you can monitor for this virus in populations of bats, and go out to sample other wildlife and see if it has spilled from bats into weasels or cats or some other thing out in the wild that the bats might interact with.
Yeah.
Do some field biology.
So
Well,
100, you accumulate enough of this kind of information, okay? And 100 years from now, if we're still around, you take your tricorder out to a cave and you scan the bats and you find out whether there's anything there that could potentially cause transmissible human infections. And that's not crazy. We can solve three structures at 3 angstroms overnight now.
Yes.
Okay. This stuff it's not crazy.
Where were the bats that were sampled in this study? Do you know?
They're South and Southeast China. So that's
Southeast
all,
Asia.
it's where a lot of these other uh, uh, CoV-2s come from, or coronaviruses come from.
Yeah, this Pipistrellus abramus,
Is the bat.
southern and Southeast Asia.
But it's not Rhinolophus, which is the one
No.
where...
No, different
Different
bat.
So I just want to add two more things from the guest essay, and you can think about how feasible these are going to be in a world that's not completely united behind all kinds of public health things. But besides the WHO, there should be agencies inside and outside of government that fund this work that should require proof that the investigators meet the global standards. Okay, so how well is that going to work in the various countries that we're thinking about? And then scientific journals should have similar standards for the studies that they accept. And then you can think about, journals wanting to have the flashy, shiny papers, and are they going to put up roadblocks? So it's a problem. But
yeah. This is a Cell paper, by the way. That's a big deal.
Yep.
They would never turn it down. They want the publicity. Oh boy. So, I think surveillance would be a great idea to see where this is and whether it's infected anything else. Yeah.
I have another little thought here. We spent a lot of time at the top of this show talking about problems with the polio vaccine. Now we're spending a lot of time talking about problems with containment and biosafety. Okay, we actually talk about these things. Okay, we don't just say, "Oh, all vaccines are fine." We don't just say you can go ahead and do any work you want to. Okay, we think about this stuff. Okay, and have real discussions.
Yep. What is it called? Making polarized statements. We don't do that.
No.
We just base what we say on data. And speaking of data, we have another paper.
We do.
Which is pretty cool. Alan, can you summarize it?
Sure. So this paper is innate immune sensing of rotavirus by intestinal epithelial cells leads to diarrhea. I always like a paper with diarrhea in the
Absolutely.
my attention.
And I can never spell diarrhea. I will never be able to spell diarrhea.
And I also want to point out, so this is an open access paper, and the graphical abstract is wonderful. I looked at it as, oh, that's perfect. They've got a picture of a little diagram of a mouse on a toilet. Anyway, so whichever one of the authors did that, good job.
Yeah, I was thinking this is because they're probably thinking, how can we show diarrhea?
We show mouse diarrhea.
Yeah.
Yes.
Yeah.
Yeah, so the mice are sitting on the toilet. The first author is Gaopeng Hao. The three co-second authors are Juhi Sun, Maria Florencia Gomez Castro, and Takehiro Kawagishi. And the senior author is Xuyan Ding. The team is from Washington University School of Medicine in St. Louis, Osaka University in Japan, University of Science and Technology of China in Hefei, University of Pittsburgh School of Medicine, and Stanford University School of Medicine. So in true international collaboration. Quick summary, they wanted to look at pathogenesis of diarrhea in viral infection, which is an enormously important thing worldwide. It one of the leading causes of death for kids. So they did an experiment that I'm baffled was not done 100 years ago. They gave newborn mice inactivated rotavirus. They inactivated it with UV light, gave the inactivated virus to the newborn mice, and found that it induced diarrhea. This is a non-replicating virus, so just the virus particles themselves are induced diarrhea. Then they dug into the mechanism, and there's a whole bunch of work here, the upshot of which is that they find the innate immune system of the gut uses interferon lambda signaling to reduce water absorption in the gut, and that's what leads to the diarrhea. Then they also did experiments in human organoids and also with reovirus to confirm it's not the unique rotavirus and mouse combination thing. And taken together, this all suggests diarrhea could be an active host defense, and it also shows for the first time that type 3 interferons can induce diarrhea. So besides showing an unknown role for previously unknown role for diarrhea in viral pathogenesis, can be part of the host response, this might help explain some aspects of inflammatory bowel disease, which is, based on inflammation, and gastrointestinal bacterial infections.
So the idea is the diarrhea flushes out the virus.
That's right.
Yes.
That's right.
The innate
Yeah,
immune
it
system
flushes.
somehow is sensing the virus and says, let's get rid of this,
Yep.
and empties
Now-
the intestine.
Rotaviruses, big cause of gastroenteritis globally, right?
Yeah.
They also make a protein, NSP4, that we talked about a few weeks ago in terms of calcium waves, right?
Yep.
And that alone, sorry,
It's TWiV 1189.
yeah,
Yep.
Viruses making waves. And that protein on its own will cause diarrhea in host animals. So when the virus replicates, it makes the protein, and that drives diarrhea. So there's something else going on here.
In this case, the virus is not replicating,
Yeah.
not making that protein, and it causes diarrhea.
Yeah, so they had this idea that maybe diarrhea is a flushing process, a mechanical process that gets rid of pathogens, and so it would be a host-driven response. But it's always going to be a mixture, right? Because when you are infected by rotavirus, right, you're going to get some of this flushing initiated by the mechanism we're going to talk about, but also the virus is going to make the enterotoxin, and you're going to get diarrhea as well.
There is always, and this is real, in terms of scientific method, this is a tough one, and has always been a tough one for me. When there's a problem, it's very difficult to avoid looking for a single solution to the problem, okay? When in fact, quite often, it's multiple things going on.
All right, so they start by saying, all right, we're going to use an activated rotavirus, so we don't have to worry about any virus-encoded factors that may be contributed. And also, we're going to focus on interferon lambda. So type 1 interferons have been used for therapy before. They cause a lot of side effects, and they say, well, type 3 interferons don't, and so maybe that's a good one to look at. And so that's what they do, and they find that interferon lambda is a big player in here. They say this is a very, very nice sentence. This study reframes viral diarrhea from a solely pathological symptom to an active host defense strategy.
So I have a little bit of trouble with this. From the jump from the inactivated virus induces diarrhea to the notion that the diarrhea basically gives you a selective advantage in a way.
Yeah.
To me, that's a pretty big leap. But I suppose the connection is that it's an innate immune response.
Yes.
It's the idea that the organism is responding to something that it doesn't yet know is actually pathogenic by inducing diarrhea.
It's treating the virus as a toxin
Yeah.
when the virus hasn't done anything yet.
Yeah.
But also, this enterotoxin, NSP4, which induces diarrhea, what's the idea there? I mean, it's just going to flush you out unless it's a side effect that wasn't intended.
Well, what I've always thought is that if you were a virus that was going to infect the GI system, what better way to get around than induce diarrhea? So maybe it's a little of both, okay?
Yeah.
You benefit by flushing this thing out. The virus benefits by getting flushed out.
Yeah.
So basically, you have to hydrate people who are having diarrhea, otherwise they can die, right? Especially infants. But the strategy of preventing diarrhea may not be the best thing you can do.
Yeah, good point.
Because we all want to prevent diarrhea because it's nasty. But, you could take Imodium, right? That's one of the anti-diarrhea things, but it may not be the right thing. And they
So
talk
you're
at the
better
end
off.
about this.
So you're better off hydrating people.
Yep.
Okay.
Yeah, hydrating and let it rip.
Well, so what I liked about this was, in that first paragraph, they made the analogy to sneezing and coughing, clearing irritants,
Yeah.
and so that's good from the standpoint of the host. Let's just get it out. But then from the standpoint of the microbe, it's a pathway to elsewhere.
Hey, it's a win-win situation.
Yeah,
that's right.
everybody comes out ahead with diarrhea.
Yeah. Well, okay. So what they do is they take inactivated rotavirus, they inactivate it with sorrelin ultraviolet cross-linking, and they infect 5-day-old neonatal mice, and they have as a control infectious reovirus.
The reason they're doing this with newborn mice is because, like humans, rotavirus is most likely to cause severe diarrhea in the newborns.
So
yeah,
infectious,
and after a short period of time, it doesn't, right?
Right.
So
yeah. So infectious rotavirus causes diarrhea in 100% of the animals by 1 or 2 days post-infection. And also 100% of the animals who get inactivated virus at days 2 and 3, right? So inactivated virus is able to make diarrhea. And they make this good point. Viral replication is needed for shedding, but not for diarrhea induction. So they figure a diarrhea dose 50%, a DD50.
DD50, yes.
For infectious viruses, 10,000 PFU, and for inactivated, it was 100,000 PFU, 10 times more. So then they try another virus. They try reovirus, a mammalian orthoreovirus, which is suspected to cause some diseases in humans, including diarrhea. And they feed this to mice. And the infectious virus causes diarrhea, and so does the inactivated form. So 2 different viruses. They're both reoviruses, but rotavirus and mammalian orthoreovirus do the same thing. So that's interesting. Right. So what's going on here? So they have some genetically modified mice that they are going to use. They have mice that lack the interferon alpha receptor and the interferon gamma receptor. And the inactivated virus causes, again, 100%, up to 100% diarrhea in wild type and those knockout mice. All right, interferon alpha and gamma receptor knockout mice. But if you knock out STAT1, which is a signaling protein involved in all the interferons, they get less diarrhea. So they say this suggests to us that interferon lambda signaling is contributing to diarrhea. So indeed, they have interferon lambda receptor knockout mice, and they don't get diarrhea in mice inactivated with inoculated with inactivated virus.
Okay, I got to interrupt here for just a second.
Yes, of course.
Have you guys been asking yourselves, I hope, how you measure diarrhea in a mouse? Maybe you already know, Kathy. You're a mouse person. You're a mouse person too, Vincent.
Yeah,
I just looked it up.
Polio doesn't cause diarrhea.
I just looked it up.
How do you measure diarrhea?
You stick the mice in some sort of a container with filter paper or a paper towel on it.
Yeah.
Okay. And if they've got diarrhea, you get a watermark under the poop, and you measure the size of the watermark. Pretty cool.
Yeah.
Okay.
Quantitative.
Yeah.
I love it.
Yeah, the thing mice don't do is they don't vomit.
Right.
Right.
That's
Good.
cool, but-
We wouldn't want that complicating.
Oh, yeah,
result.
right. All right, so looks like interferon lambda is somehow involved here. And they look at the timing of this. So they look at the mRNA of interferon lambda, and they find the mRNAs go up maximally at 8 days post-infection. They look along the GI tract. It's very high in after inactivated rotavirus administration. It's very high in small intestinal tissues. But there's also some in the large intestine, but much less induction over there. And of course, the small intestine is mainly where virus reproduction occurs here with rotaviruses. And they look for the protein interferon lambda. It's in the ileal epithelium, but not in the lamina propria or the distal colon. Okay. So the lamina propria is below the epithelium, right?
Yep. Between the epithelium and
Now,
the basement membrane of the muscle.
if you're thinking interferon lambda is important for inducing diarrhea, then the obvious experiment is to feed the mice interferon lambda, right? Because that's what was done with NSP4. They fed calves with the protein and they got diarrhea. So they get recombinant interferon lambda. They give it intraperitoneally into mice, and it induces diarrhea in up to 60% of mice. So, and interferon alpha doesn't do that. Okay. And they do this at different mouse facilities in California, St. Louis, and Sweden. And they say it shows that the facility microbiome, which differs in the mouse from facility to facility, is not really making a difference here. So interferon lambda signaling is needed for diarrhea occurrence in neonatal mice. They also do some more transcriptional analysis by looking at, so the way interferons work is they bind to receptors on cells and then they induce the production of what we call interferon stimulated genes, or the transcription of ISGs. So they look at a canonical ISG15 just to survey across the tract, right? Because you're making lambda, and then where is its effect seen? And they see ISG across the GI tract. Remember, lambda was mainly in the small intestine, but now ISG15 is across the tract. And so they're thinking it's,
sending a signal downstream.
Sending a signal downstream. That's right. They also
downstream in the true sense in this case.
They do a cool experiment where they can selectively delete the interferon lambda receptor in intestinal epithelial cells. And so they have these mice, and when you infect them with the inactivated virus, there's no diarrhea. So small intestine lambda, interferon lambda, acts locally and distally to mediate the diarrheal response. All right, so that's all mice. What about cells? We have these human intestinal epithelial cell cultures. They have donor-derived human ileal organoids. So obviously these come from a donor, and you grow them in culture, and they make structures that look a bit like the gut. And in fact, if you use those in these experiments, the inactivated rotavirus gives you high levels of interferon lambda secretion. So then they delete a variety of, they want to know what's the signaling. So interferons respond to sensing. There are sensors of all sorts within the cell that sense things like double-stranded RNA or protein, and they turn on interferon transcription. So they want to know which of the sensors. So they make a whole bunch of knockouts, RIG-I, MDA5, MAVs, all these proteins involved in the sensing of double-stranded RNA. And they do this in a human cell line. They find that the induction of lambda is attenuated in the RIG-I knockout. So RIG-I is involved in the sensing of RNA. And it's also abolished in RIG-I MDA5 knockout. So both RIG-I and MDA5 are involved in sensing RNA. So they think it's cytosolic double-stranded RNA that is being sensed in the cell, and that's responsible for interferon lambda production. So
now the inactivated virus they put in, they can't replicate, but the virus is intact. It's got its genome still, right?
Yeah. So that's a really good question that I've been thinking about, right? These rotaviruses have double icosahedral shells, right? And in fact, during infection, the RNA never leaves the shell. It's transcribed, it's copied into mRNA within the shell, and then the mRNA comes out. And that's what the outer shell missing. And I always viewed that as an immune evasive strategy, you keep the RNA in the shell so it can't be sensed. But
Hide
here,
the RNA. Yeah.
you hide the RNA, but here it's something's
How are
going
these
on.
RNA sensors see?
Yeah, I don't know.
Well, nothing's 100%.
Yeah, some of the virus
Yeah.
may break, right? Yeah. Yeah, it's too bad. I really like things to be. But yeah, some of the
Sorry,
particles
Vincent.
could break open. Someone in class the other day said, asked me this question. I said, I think this sensing of rotavirus RNA is the keeping of rotavirus RNA in a particle is a antagonism mechanism. Anyway, they also have mice that lack the MAVS gene, which is a downstream protein, which encodes a downstream protein in the RIG-I MDA5 axis. And fewer of those mice get diarrhea when they're inoculated with that inactivated rotavirus. So that tells me there's probably some other sensing things going on as well, because it's not 100%. All right, so they think viral RNA is the, so this is called a PAMP, a pathogen-associated molecular pattern, like it could be RNA or DNA or a protein.
And these are things that typically trigger the innate immune system.
Right. Now, Margaret McFull-Nye.
So it's hard out there for a PAMP.
Margaret McFull-Nye years ago made the suggestion we should call them MAMPs because it doesn't have to be a pathogen to be sensed. It could be a microbial, microbial associated molecular pattern. I like MAMP better, but
that's good.
Right?
I like that.
That makes a lot of sense. So they use empty particles of rotavirus, and those do not induce diarrhea they don't induce lambda expression in cells. They don't induce diarrhea in mice. If they incubate these inactivated rotaviruses with neutralizing antibodies, you don't get any induction of interferon lambda, and there's no diarrhea. So it says the virus must enter the host, right? And then this is a cool experiment. So you can buy double-stranded RNA. It's called poly-IC, right? Synthetic, and people use it all the time to induce interferon responses because it's sensed by this sensing pathway. So they inject poly-IC into mice. 86% of the mice have diarrhea. And if you do that in interferon lambda receptor mice, no signs of watery diarrhea. So now this term has just come into play here, watery diarrhea. A single-stranded synthetic RNA.
So maybe they had some diarrhea, but it didn't score as high?
I don't know. They all of a sudden started talking about watery diarrhea.
Hmm.
Single-stranded RNA doesn't do this. If they infect the mice with a murine...
And it's a good thing single-stranded RNA doesn't do that.
Yeah, a messenger RNA is...
Because we use that.
Yeah, that would be a problem. Our cells are full of single-stranded RNA.
We'd be up a creek, so to speak.
And they are always breaking open in the intestine, so I'm sure that would be a problem. Murine neuroviruses make double-stranded RNA intermediates. They, when you infect mice, it induces diarrhea. And they say have increased water content compared with interferon lambda receptor negative mice. Anyway, it's a different virus doing the same thing.
And it's a single-stranded genome, so
yeah, right.
You got to get into replication. I don't think you can really do the inactivated virus experiment.
No, that's right. So there they use infectious virus, and they're making double-stranded intermediates. Although I could argue that it's a viral protein that's doing it.
Yeah, because noroviruses make a toxin as well, don't they?
I don't think so. It's been identified, but you can imagine that there would be a protein that could do it. So that's a little iffy, that experiment. Okay, how does it work? So they look at serotonin secretion by enteroendocrine cells, and that's triggered by rotavirus infection. They do a series of experiments to and basically show that serotonin doesn't seem to be involved in the diarrhea. Serotonin is not the primary driver of diarrhea. They also have They look at gut motility, and that doesn't seem to be altered by rotavirus. They look at intestinal permeability. It doesn't seem to be changed. The stem cell differentiation in the gut doesn't seem to be changed. But what they do find is that one of the proteins in the intestine that's important for fluid balance, its levels are changed. They do a single cell sequencing experiment, and they find so following the rotavirus infection of mice, they find decreased transcripts of a protein called GRA, which is involved in exchanging water with. So it's usually some other ion that it gets exchanged with. And so essentially they're thinking that the downregulation of DRA by interferon lambda is causing water release from the intestinal cells. They can actually duplicate this in cells in culture. They can see dropping of DRA mRNA by adding interferon lambda. So they look more at DRA in diarrhea. They show that in mice, putting in an activated rotavirus causes downregulation of DRA. Their DRA is not changed in interferon lambda receptor knockout mice, so that tells them that lambda signaling is upstream of DRA. They make ileal organoids and show that you can put interferon lambda in those, and it decreases DRA. And if you knock out the interferon lambda receptor in those cells, DRA doesn't go down. They're also, fortunately for them, there's a DRA inhibitor. When you give mice the DRA inhibitor, they get diarrhea. My God, this is a perfect experiment. Right?
Nice, works nicely.
It comes full circle. Now, this is interesting. When they give mice interferon lambda, remember they develop diarrhea. If you give them DRA, they don't get any worse diarrhea, right? And you can measure the worseness of diarrhea apparently in different ways.
Well, as Rich described, you can measure the amount of water that soaks
fecal
into
water content, fecal scores, and diarrhea incidence. 3 things. So that says they're in the same pathway, and interferon lambda recapitulates DRA, right? It has the same effect. What about humans? So they have colonic monolayers, they're polarized, and they can show that if you give these cells, they're called colonoids. You give them inactivated rotavirus, interferon lambda, or DRA inhibitor, they have a problem in keeping water inside of the cells. It's very cool. You have the cells on a membrane with 2 chambers, and you measure the volume in the upper chamber when you do these treatments. And so if you add inactivated rotavirus or interferon lambda or DRA, you get reduced fluid in the upper chamber. It's very cool.
There are reduced or increased fluid?
Reduced in the upper chamber, yeah. I'm not going to think through it right now. That's just the result.
Right.
Then finally, last experiment. Let me just go back. I want to figure this out. Okay, so you have monolayers in transwells.
Right.
So my understanding is the cells go on the top of the membrane, right?
Right.
And then it's permeable, so there's medium underneath it as well. And so they say the media volume in the apical compartment serves as a surrogate for apical ion activities and reduces as anion transporters such as DRA are activated. Okay, so yeah, if you activate DRA, it pulls water into the cells, and so it's going away from the upper chamber. The problem with DRA is when you inhibit it.
Like right. They activate. Got it. Okay, so
Yeah.
you activate draw and it pulls the
Yeah.
water in. You deactivate draw, which is what happens with the
Yeah.
rotavirus,
Exactly.
and that causes the water to go out.
Yeah.
And that's going to be diarrhea.
And finally, they use primary human intestinal organoids from either healthy people or people with IBD, inflammatory bowel disease. And they noticed that the levels of interferon lambda transcripts are really much higher in these IBD organoids, so tenfold higher. And they also look at people with either ulcerative colitis or Crohn's disease, and where they find similar findings with interferon lambda. And biopsies from people with ulcerative colitis had decreased DRA expression compared with healthy controls. So it's just a few observations there which are tantalizing because, these individuals can have diarrhea and maybe there's some problem with lambda regulation, and that's why they're having it in part.
Yeah, because they found, in this paper, they're finding what's apparently an innate immune mechanism that induces diarrhea in the presence of a threat.
Yeah.
And now they're looking at people who have various immune disorders that affect the gut, and they're seeing that those disorders dysregulate a similar pathway or the same pathway.
Yep. So there you go. Rotavirus is sensed. It induces interferon lambda signaling. So the viral double-stranded RNA is sensed. You get interferon lambda made, which downregulates this apical chloride exchange, or DRA, and you get increased intestinal fluid secretion. That's one mechanism for diarrhea. Remember, there's another also, which is caused by the viral protein NSP4. I thought this was very interesting.
So the next time that you're laid out flat with diarrhea because of some viral infection, you just think happy thoughts. Okay.
Oh, I'll know what's going on,
for
right?
you. Yeah.
I'll know what's going on. So then the thing is, what's the therapeutic implication of this, right? And I couldn't really suss from their discussion what they were thinking. But they say, depending on the context, therapeutic enhancement or inhibition of interferon lambda and DRA could be beneficial in conditions characterized by dysregulated intestinal electrolyte and fluid balance. So they're being non-committal.
I think we need to know more about the systems in question before we start, agonizing or antagonizing DRA.
It's nice to know the mechanism. It's really interesting.
And this is a step in that direction.
And maybe it's applicable to other viruses that cause diarrhea as well. We need to find that out. But don't go out and get a DRA inhibitor would just give you more diarrhea.
Yes.
You would need a stimulator of DRA. But you don't want to do it. You want it to flush out the virus. That's the whole point, right?
That's, to me, the biggest therapeutic implication.
Cool. By the way, I noticed that our friend Terry Dermody is on this paper.
Mm-hmm.
He's got to be the real virus component in all of this.
Yes. Okay. Science moves on. Let's do some picks of the week. Kathy, what do you have for us?
Well, when we talked about ASV, I meant to have it trigger. I wanted to just mention a couple other things about Anne Palmenberg, who passed away, and you guys discussed it on a previous episode. I interacted with her a lot. She had been president of ASV. She'd been the local host at probably almost every single ASV that it was ever at Madison, which is a lot of them. She just was very, very helpful to ASV from the early days and onward. And then she also put together, along with Rob Kaleda, a database of several different types of meetings, the herpes virus meetings, the ASV, and a couple others, how many women speakers there were. And they published papers about that. And the numbers of women speakers in the plenary sessions and so forth, I think, at least in the virology-related areas, has gone up, but it was something that she was very passionate about. And because of all of her work for ASV and also all of her contributions to science, she was the recipient of the American Society for Virology Yaklik Award. And so I just wanted to say that for me, she always had kind words. She was a very hard worker. She was very active in things. And then some little fun fact that people might not know, and that is that when she would use the abbreviation LOL, she was pretty consistent about thinking that it meant lots of love. So if you go back and you look at any of your texts or emails or something where she put LOL, she wasn't saying laughing out loud. She was sending you lots of love. So I think that was pretty cool. So the two picks I had just kind of stood out to me. They came up, both of them came up in the Science Advisor. Two things that mice do. One is that it's been discovered that the most commonly used strain of mice, C57 Black 6 in laboratories, will cry out when they urinate. But and so it's still
ultrasonic, right?
Yeah. It's still only a preprint, and the significance of these vocalized urination behaviors is still unclear. They raise the possibility that maybe it's strain specific because they looked at a mixed background mice with C57 Black 6 crossed with another strain, and it doesn't seem to happen. But they looked at a lot of factors and they can't quite figure it out. So who knows what that's about. And then the other one is a paper that's published in Science, and that's showing that mice give basically a mouth-to-mouth resuscitation to unresponsive mice. And so there's a short video and a little picture in this link that I put in. So both of these are the links to the Science Advisor, and then you need to scroll down until you find the mouse picture. But the mice will start with sniffing and then grooming the unresponsive mouse, and then they will do a very intensive physical interaction. They kind of pull the tongue out, and they actually show him like with his paws on the mouth and giving mouth-to-mouth resuscitation.
It's amazing.
Mouth to mouse.
Yeah. So I thought that was interesting. And then, unfortunately, I heard this, and then I kind of checked it online, that evidently Trump made some claim about how many millions of dollars the NIH had spent on scientists making transgender mice.
Oh God.
And
oh no.
Instead of transgenic mice. Yes. So
wow.
Yeah. So anyway, so that's another thing, ignorant people just there's a difference.
The stupid burns. Wow.
Yes. And so then also these friends of mine, we were out to dinner and they said, and people are going to have to worry, chemists, biologists, so forth, about putting the word cis or trans in their grants. Yeah. Because
I've been restraining myself the whole episode from referring to transgender mice.
Yeah.
We've been talking about mouse experiments.
Yeah. Yeah. But the studies here used transgenic mice, which is completely different thing.
Totally unrelated.
Yeah.
Sad, very sad. Rich, what do you have for us?
Well, those of you who pay any attention at all will know that there was recently a soft landing on the moon by a commercial enterprise, the first of its kind. And so I thought I would pick, for reasons that'll become obvious, the company that did this. They're called Firefly Aerospace. And you can look through their website. I think these commercial space enterprises are fascinating. And if you look through what they want to do everything, okay? They want to go up and service stuff. They want to transport stuff to the moon. They want to do orbital flights of one sort or another. And this lunar landing is, they've got a bunch of NASA experiments on board that they want to do. But among other things, it's just proof of concept that they can do it.
Yeah.
One of the things I like is that these dudes are in Cedar Park, Texas, which is just about 40 minutes north of here. Okay.
And they're a contractor. that will put your stuff in orbit or on the moon. And this particular mission, NASA hired them to land experiments on the moon, and they did it. And there are one or two other companies that are doing a similar thing. One of them, I think they haven't been able to confirm that their lander touched down, or they hadn't last time I checked the news, and then the other one may be on the way.
They have a cool video up the top of this that basically is video from the spacecraft that shows the whole descent and landing. It's pretty cool.
It's a cool looking lander.
Yeah.
Very neat.
There's more I want to know about. I always want to know about all of the intermediate steps in something like this, because the lunar landings for the Apollo mission were extraordinarily complex in terms of what vehicle was contained in what other vehicle and how many stages there were and that kind of stuff. They don't really show me everything I would like to know on this, but still, it's very cool. Good on them.
They do have the advantage that they don't have to worry about getting anybody back.
True.
Yeah.
Yep.
Which was a big part of the Apollo missions. You had to have the capsule orbiting, and you had to be able to relaunch from the surface of the moon and get back to it, and then get all the way back. But these robots don't realize it's a one-way trip.
Speaking of space, another SpaceX rocket exploded.
Yep.
He said, no big deal, kind of like measles is no big deal. Boy, would you notice a trend here? Huh?
Yeah, well, yeah, the strategy here is just, give it a try. And if things blow up, you learn something.
Yeah, okay. Alan, what do you have for us?
I don't want to be one of the things that gets blown up.
No.
Thanks.
It's fine if you're doing hardware, not so good if you're doing people. So this is a delightful place that Laura and I visited about a month ago. She was actually working in Buffalo, 6 hours away, which, she's done. She's back. Very happy about that. But right out in Rochester, in upstate New York, there is the Strong National Museum of Play. And
that's cool.
I saw this. It popped up in some kind of search for things to do in the Buffalo area, and Rochester's only an hour away from there. And the more I looked at it, the more I said, oh, we got to go check this out. It's nominally a toy and game museum, but they take their mission in a very broad sense. It's about the human urge to play in general. And it's amazing. You'd think, okay, it's a toy and game museum, it's for kids, but it really is something where no matter how old you are, you can go there and see your childhood. I mean, they've got everything. They've got the whole history of toys and games going back to whenever. And they've got a bunch of interactive stuff you can do. There's a ropes course you can go, climbing up on. We did that. There's a butterfly garden. There's, just a bunch of interactive displays. I took a selfie on the Sesame Street, an old Sesame Street set. They've got that set up there with, Big Bird looking out a window. So if you are anywhere near upstate New York in the Rochester area, definitely swing by here. It's a blast.
Looks great.
Yeah.
Why is it called the Strong?
It's named after somebody who donated the initial collection.
There's I don't know who Strong was, but there's Strong Memorial Hospital as well.
Right.
He's a big name in Rochester.
That's right. Excuse me. My pick is an article in Nature by Humberto Basilio. It's called How a PhD Student's Lab Size Affects Their Chance of Future Academic Success. So this was a study which is published. You can go look at the original paper where they talked to labs around the world, and 1.5 million scientists, 1.8 million mentorships, chemistry, physics, and neuroscience. And they concluded that postdocs, grad students, and other junior scientists in large research groups are more likely to drop out of academia than their peers in smaller groups. But when they stay, they have more career success than people in smaller groups,
Whatever doesn't kill you makes you stronger.
Yes.
I guess.
Or just
I
gives
don't
you
know.
diarrhea.
I got my PhD in a small research group. I was fine. Peter Palazzi had just started out, so. But that's an N equals 1. So there you go. This is interesting. The one of the authors of the study, co-author Roberta Sinatra. I hope this helps guide the choices of PhD students, which I guess she's saying you should go work in a big lab, huh?
Well, yeah,
I don't
I mean,
know.
if
you want to
you'll
survive,
either burn out or you'll do well.
Yeah.
Mhm.
I always
I'm
like the
sure-
small. I always like the small lab environment.
Yeah, and a big part of this is that who has the big labs? The people who have the big labs are the big names who have the big grants, and they're at
Yeah.
the big institutions, and so you're connected to all that, and you're accustomed to doing research in that environment, and of course you're probably going to go on to a postdoc at some similar high-profile place, and yeah.
The other thing that I think of is that, we have a tendency all the time to think of this in terms of labs and research and that kind of stuff. But these people are all university professors. There's more to it than just papers and that kind of stuff. You're an educator. And matter of fact, one of the things that used to just really annoy me was people who were so into their research that they didn't give a hoot about education or mentorship. Okay.
Mm.
And a lot of when I look at this graph and I see the people from small laboratories becoming science survivors, I see them running small laboratories and teaching and being university professors and keeping the whole thing going. All right. So good on you.
Or following the path that the majority of PhD students do and not pursuing an academic career and doing something outside the tenure track academic career.
Right.
One of the factors they cite here is that the people who are in the big labs make papers that are more highly cited than the people in the small labs. They have a higher citation index.
But does this look at like every person from that big lab? I mean, wouldn't there be some of those people who aren't, they're going to go on and maybe not get the high profile big name institution position, and they're going to be at a smaller place.
Yeah.
And are they going to thrive and know how to do the educating and mentoring that Rich was talking about? Are they going to have had role models for that? And are they going to, survive in a place that doesn't have all the same resources? Like if you trained at a Howard Hughes supported lab and then you had to go to a lab that, your own lab that didn't have those kind of resources, would you be able to?
I don't know the answer.
Frugal. Yeah.
Anyway, I thought I wanted to be a teacher. Okay. Yeah. And I liked the lab, and I thought that was kind of a bonus. Right. I didn't realize until later on that was kind of flipped, at least in the minds of some. Mm-hmm. And I just want to kind of emphasize that being a university professor and a teacher is an important job.
Yeah. It's open access, so you can check it out.
Cool.
Listener pick from Larry TWiV. My pick is this Washington Post article about a super antibody carrier. One person can make a big difference. So this is behind a paywall. But the headline is James Harrison, whose rare blood protected millions of baby dies at 88. A rare antibody in his blood was used to create 3 million doses of an injection needed to protect newborns. And I can't read any further than that. Does anybody?
Yeah.
I don't have Washington Post.
I do.
Go ahead, Kathy.
Yeah, I don't have it up, but I can do it.
And if I were a subscriber, I'd cancel at this point anyway. But
yeah, that's what someone I just sent a gift link of some article in the WAPO said.
So it contains anti-D an uncommon antibody which could be administered during pregnancy to help prevent hemolytic disease of the newborn, HDN, which can cause serious complications, including stillbirth and neonatal death.
Yeah.
So it's I guess, a rare but important condition that was treated with his antibody. I would think that nowadays you'd be able to make a monoclonal.
Yeah,
that
or
would do the same
monoclonal
thing.
cocktail.
They still administer it in about 70% of pregnancies in Australia every year.
No. Yeah, maybe you could get the sequence and heavy and light chains and just make it, right?
Yeah.
He earned himself the moniker, the man with the golden arm.
Thank you.
HDN arises when a
Larry.
woman with an Rh negative blood type carries a fetus that inherited the Rh positive blood. Okay, so this is the whole Rh thing.
Yeah.
Okay.
Cool. Thank you, Larry. That's TWiV. $11.99. You can find the show notes at microbe.tv/TWiV. If you want to send in a question, a comment, or a pick of the week, please send them to TWiV at microbe.tv. If you like these programs, we'd love your support. Microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Conda is an Emeritus Professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Thanks.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamp. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.